Hi, everybody.
Hi there.
Hey. So thanks for coming. Thanks for the opportunity to talk, Vince. So as Vince said, I'm going to talk a little bit about methylation sequencing, the technologies, the kind of methods that are available to you if this is something that you want to do. Hopefully I'll show you that it's quite a complicated space, okay? And really what we're trying to do is simplify and really streamline. So Cambridge Epigenetics is a company that was founded in 2012. Okay, we were spun out of the University of Cambridge on the back of a patent for oxidative bisulfite technology. Okay, so I'll explain exactly what that is in more detail later. But we were founded by Shankar Balasubramaniam. Does anybody know who Shankar is? One guy. One guy. Yeah, you at Cambridge, right? So for those that don't know, Shankar held most of the original patents for the SBS sequencing by synthesis technology that was put into Solexa and later bought by Illumina. So all of the tech that's in Illumina's machines is basically Shankar's. Okay? So he found CEGX, Cambridge Epigenetics, we abbreviate to CEGX, to do oxidative bisulfite initially. Okay? So we are developing lots of things in R&D all of the time. We've got a new library prep method that we've developed in collaboration with somebody, which I'll share some data on later. But for now, I'll give a bit of background about epigenetics. Okay? So what is it? Well, this guy, Conrad Waddington, he defined it in 1942, I think. No, he coined the term in 1942, but he defined it in 1959 as the branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. So I think what he's saying is that anything that we can't describe using genetic principles, classic genetic principles, is basically epigenetics, okay? And he's quite famous for proposing this classical epigenetic landscape where you've got the fate of a cell, let's say, and the cell with one genotype can flow down this landscape, take any one of these paths and become a different cell, okay? So as the field evolved, we kind of learned what the units of epigenetics were. So we're looking really at modifications of histones, of DNA, of RNA, and microRNAs are involved as well. So histone modifications tend to kind of open and close the chromatin, so it's accessible or inaccessible. DNA modifications turn on or off gene transcription. RNA modifications and microRNAs are post-transcriptional regulation, so maybe those RNA transcripts are translated or not. So why does it matter? Why does epigenetics matter? Well, as I kind of hinted at before, stem cells can differentiate into a whole multitude of different cells from one genotype, but they're epigenetically distinct. Okay? Aberrant DNA methylation can serve as an early detector of cancer. Identical twins can be discordant for disease risk, but because their epigenomes are distinct. And it may have a heritable and non-heritable influence in complex disease development. Okay? So as a field, it's expanding a lot. You know, these number of publications, kind of all of the omics, the genomics, transcriptomics, they all follow this trend. I've marked what most people classify as the first NGS system on here, 2005, the 454. But the numbers involved really are not massive yet. All right, so genomics and transcriptomics are generally bigger fields. And there are several reasons for this. Really, it is quite hard to do methylation sequencing. The analysis is difficult. It's difficult to get good quality data. And I'm going to show you why the classical approaches really are not sufficient or not good enough. So I'll talk now about the different DNA modifications and how we can sequence them and study them. So in 1948, it's a long time ago, 5-methyl-cytosine was discovered or described. So C could exist, but it could also exist in a modified form. And later, in 2008, here at Cold Spring Harbor actually, an epigenetic trait was defined as a stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence. Okay? But then, a year later, in 2009, things got a little bit more complicated. So, in 2009, these TET, these 1011 translocation enzymes, were discovered. And in parallel, more modifications of C were discovered. So the TET enzymes will oxidize 5-MC to HMC, and then HMC to FC, which is 4-methylC. This is hydroxymethyl-C, 4-methylC, and carboxy-C. Okay? So I'm going to refer to these as 5-MC, 5-HMC, 5-FC, and 5-CAC. As you go down this pathway, the levels of each one decreases by roughly an order of magnitude. So we're not really able to look at carboxy-C yet. Okay, the amounts in the epigenome are so small, the techniques are not sensitive enough, we'd have to sequence very deeply, so it'd be very expensive to do. But we've got the capability of analyzing these two. Okay? So how do we do that? Well, bisulfite sequencing was introduced in '94 by Sue Clark. And at that time, there was only C and MC. The other modifications weren't known. So it was developed to distinguish between the two forms. So you expose DNA to bisulfite, and you convert any unmethylated Cs to uracil. They deaminate to uracil. And any methylated cytosines are resistant to change. Okay? Some of them can change. Yes?
Can you see the difference between 5-MC and 5-HMC?
Yeah, I'm coming to that. So some of these can convert, okay, to U. It's a very slow reaction, but they are very resistant to change. So when we sequence and we see a change in the sequence, we know that that is because it was an unmethylated cytosine. Okay? That's how the field was. But then all of these different modifications were discovered, and in 2010 it was shown that they all behave differently under bisulfite. So HMC will go to CMS, which is cytomethionine sulfonate, I think. 5-SC goes to U, and carboxy also goes to U. So now we've lost the ability to distinguish between the different modified forms. Okay? So again, you convert C to U, MC resists change, 5-HMC to CMS, and 5-SC to U. Now when you sequence these, U is read as T, 5-MC is C, CMS is C, and U is T. So actually bisulfite data is mixed. Okay, you have a mixture of methyl-C and hydroxymethyl-C. So what Shankar and his group did is they developed an oxidation method that's upstream of the bisulfite conversion. So it specifically oxidizes the OH group, and 5-HMC is oxidized to 5-FC. So now, when we bisulfite convert, and sequence, all we're really reading are the methyl Cs. Okay, we get a true measure of 5-methyl-C. Okay. One of the challenges developing this technique was that 5-SC converts to U, and it's quite a slow reaction. Okay. So because we're generating more FC than is naturally occurring, we had to optimize the bisulfite conversion conditions to make sure that we convert all of this extra 5-SC to U, so we get a clean signal for methyl-C. However, if you're too harsh, then you'll generate or you'll convert MC to U. So we also had to be very careful not to over-convert MC into U. Okay, so that was one of the biggest challenges that we faced, but we overcame it. So OXBS, as we call it, allows you to detect just MC. If you combine that data, or if you're interested in looking specifically at HMC, you can combine OXBS with BS. And because OXBS is just 5MC, and BS is 5MC plus 5HMC, when you subtract the data, you can actually get 5HMC. Okay? So some customers also want to look at FC. So we've also developed not an oxidative bisulfite, but a reduced bisulfite, a reduction bisulfite. Okay? So we have a reductant that reduces 5FC to 5HMC. That then becomes CMS after bisulfite conversion. And when we sequence, we get all of these Cs represent modified forms of C. So that means that you take this mixed signal here, and from that you subtract the bisulfite signal, which allows you to get 5HMC. Okay? All of these reactions are at single base resolution. They're all quantitative. So we think that this is kind of our first technology that kind of brings accuracy and clarity to bisulfite sequencing. Okay? So a little bit of data to show you why it might be important to do this. This is actually data from an array, a methylation array. I'll show you sequencing data later. But if you just so these are 4 probes on the array, and if you just do bisulfite here, then you get these various levels of methylation at those specific sites. But when you do OXBS, you see something much more interesting going on. So this is true levels of methyl-C. And you see at this probe, I think it was 5% of this signal comes from MC. So when you subtract the 2 data sets, you actually see that there's a lot of HMC here. Okay, 50-50 split in these 2 probes and not much in this one. So it can really bring a lot more information to your data, or to your samples. So are these modifications important? Well, several studies have shown that HMC and FC are stable, and they're not intermediates in some kind of demethylation pathway. Sometimes they are, but sometimes they are very stable. HMC levels are highly tissue specific. So in brain, I think you can get levels as high as 70%. Okay. Also, stem cells are very high in HMC, which is interesting because usually they're found in non-proliferating tissues. Okay. You see several studies have shown that reduced levels, you see reduced HMC levels in cancer. And several studies have also shown that HMC positively associates with transcription. So classically, MC silences gene transcription. But HMC is actually positively associated with it. So it has the opposite effect in some cases of MC. So I'll now talk about sequencing and how we go about studying these DNA mods. So if you want to sequence, if you want to do methylation sequencing, you essentially have 4 options. You can perform some kind of enrichment for the DNA mods, for the DNA modifications. You can enrich and then bisulfite convert. Okay, so you can kind of combine bisulfite with some kind of enrichment. You can do whole genome oxbisulfite or bisulfite. Or you can perform some kind of targeted enrichment. Okay? So I'll go through each one of these and explain exactly what we do. So to enrich for DNA mods, the classic method really is called MEDIP. Okay, this is methylation DNA immunoprecipitation. So it's first performed in 2005, it was later applied to NGS in 2008. And then in 2011, the appropriate antibody became available to do this on HMC as well. And several groups did that in 2011. So how do we do these experiments? Well, we make a library, classic genomic library, okay? So we've got our adapters. We fragment our DNA, we DA-tail it, we ligate adapters. And some of these molecules in the library, that form the library, will contain methylated or hydroxymethylated cytosine. So we might do a MEDIP or a H-MEDIP or both. We do an antibody pull down, so we're enriching for these mods. And our final libraries, we assume, contain only molecules with modified cytosine. Okay? I say assume because one of the drawbacks of this technique is the specificity of the antibody. Okay? Some fragments you pull down may not have any modified C. So that is one of the drawbacks of this technique. It greatly reduces the amount of sequencing that you need to do, so the cost is much lower.
What's the percentage of the false pull down?
Sorry?
What's the percentage of the false pull down?
I don't know. That's a difficult thing to say because if you sequence what you pull down, you don't know exactly what's converted, what's modified or not.
Would you recommend also then doing an IgG pull down and then sequencing that?
I guess you could do.
Or just an input?
Or?
Just your input?
Yeah, that people do that as well. Yeah. So one study that was done by Jordana Bell in London. She looked at, so she did this on cases and controls. So it's a classic application of this technique is to look at DMRs, which are differentially methylated regions. So you have a number of cases, a number of controls, you make libraries for those, you pull them together, and then you pull down. And then you look at the difference in the regions that are pulled down between cases and controls. So they set this false discovery rate at 5%, and they found, I don't know if you can see them, 9 DMRs which were obviously different between their cases and controls. So they were looking actually at pain-sensitive patients. And some of these regions were in pain-sensitive genes that were of interest to them. So as a technique, it can work, of course. You can get nice data out of this. But you definitely don't get the complete picture, okay? There's no conversion, so you're not able to say exactly which cytosines are modified. You're just basically looking at regions. So the resolution is limited to your library size, your insert size of your library. So the next method you can do is some form of enrichment and then conversion. So a popular one to do is called RRBS, or reduced representation bisulfite sequencing. Now this involves or uses a restriction enzyme, which cuts as shown. And what this does is it enriches for CG-dense regions of the genome, okay? So CPG islands, or CPG sites are CG dinucleotides, which are the classic context in which cytosine is modified, okay? And dense regions of these form islands, which are also known as promoters, usually. So to do this, we take our genome. This is a small section of our genome here. We digest. Okay. And then we have, so MSP1 will always cut in the same places. All right. So all of these sequences are represented by essentially the same fragment. Okay. We don't randomly shear the DNA here. We're specifically cutting it at MSP1 sites. So what that means is some fragments will just be too big to sequence. I mean, you might be able to make a library with them, but they won't cluster properly and they won't sequence. These big ones often contain methylation in non-CPG context as well, which as we're learning is becoming more important. I've got, later I showed...
What's too big?
Too big?
Yeah.
Well, anything too big to cluster, so anything that's over probably, you know, a kb, 1.5 kb maybe.
Could you sequence it on PacBio?
Are you from PacBio? Okay. Yeah, you probably could, yeah. As long as you can make a library from that fragment, yeah. So, I see why you asked that question. So, CPA methylation, there's a study which I'll show a little bit of data on later that's actually shown this to be actually really quite important, okay? So, RRBS is great, but you will miss this, okay? So we've got our one nice fragment from that section that we can successfully sequence. So what we do, well, I should say, some of these Cs will be methylated, or some won't be, all right? Or hydroxymethylated or modified in some form. But these nice CG overhangs we can actually hijack and use to adapt the DNA, okay? So these are the adapters. We ligate them. Then we perform bisulfite conversion. All right, so these light grey lines are the converted strands. We've got the non-methylated C that's changed to a U. So that actually means now that these strands are not complementary. Okay, so one effect of converting with bisulfite is that you get single-stranded DNA. Okay. You then do a PCR to complete your library, if it comes up, yeah. So what you will actually end up with are two different products, one from the bottom strand and one from the top strand. All right, so when we sequence and when we map this back, we have to take that into consideration. So it can be quite complicated to analyze this data. So what are the disadvantages of this technique? Any given sequence is only represented in one fragment because we're specifically cutting, we're not randomly shearing. We don't get any non-CPG methylation. We will always miss some regions, although that's kind of the point because it's reduced representation. But some of those regions may be important, or some of them may be so small that you miss some CPGs. A non-complete digestion with MSP1 basically may mean that your results are not very reproducible. So that brings us on to whole genome bisulfite sequencing. which is the most expensive of the methods, but also the most informative. Okay. Yeah.
Aren't there also enzymes that you can use that are methylation sensitive or insensitive?
There are some, yeah.
And isn't that also another option that you can use?
Mhm.
Like, have you dabbled with that at all?
No, we haven't as a company, but some people do that. Yeah. I mean, I'm just kind of going over the most common techniques and applications, but there are definitely methylation sensitive enzymes. So within whole genome bisulfite sequencing, there are actually 3 options for your library. So I think maybe, hopefully, you're starting to see that it's not actually simple, right? You've got so many options. You know, if you do this, as we'll see, pre-BS libraries, you're actually going to be using tech that was developed for genomic sequencing, not bisulfite sequencing. You're buying a kit from this supplier, one from this supplier, you're kind of hacking them together. Maybe the output of one kit's not very compatible with the required input for another. So we've developed a post-BS method. I'll explain all of these individually soon. And it's an all-in-one solution. Okay, so you convert your DNA and it goes straight into your library prep. Everything is compatible, there's no mixing suppliers, it just all works well. So what do I mean by a pre-BS library? Basically, you make your library before you bisulfite convert. All right, so again, just like the MEDIP technique, classic NGS library, genomic. The only difference is that the adapters need to be methylated. So any C's, any C's in these adapters must be methylated. Why? Because when we convert, we get a change in sequence if they're not methylated. And we don't want that because it won't anneal to the flow cell, it won't sequence, the sequencing primer won't bind. So we must use methylated adapters. So as before, the DNA becomes single-stranded because it's no longer complementary. But a second thing also happens under bisulfite, which I didn't mention before, and that's that it fragments your DNA. So it's actually quite a harsh treatment, okay? And what happens, if you see, you end up breaking all of your nice library molecules, and the fragments they're not adapted at both ends anymore. They've only got one adapter attached. So when you actually look at the details and quantify how much, you basically destroy 98% of your library. There's only about 2%, roughly, that remains amplifiable. So amplifiable means that it's an intact, single-stranded library. So we take these 3 fragments, and to complete the prep, we do a PCR. And there we have our final bisulfite library. Okay, so that will sequence. So I think there are several disadvantages with this technique. It's the traditional way, it's the most common way that's done. But you need to put a large mass of DNA into the reaction because you lose about 98% of it. So you get a very low yield. You get high duplication rates because essentially you make this much library, and when you convert and amplify it, you're left with this much. So you end up with a lot of PCR duplicates in there. And high duplication rate basically means you've got a low diversity library. So I'll explain a little bit more about diverse libraries later. Have you come across this technique, this concept yet in the course? Okay.
No.
Not in the course. Okay, but elsewhere. All right. So okay, that brings us on to PBAT libraries, which stands for post-bisulfite adapter tagging. Okay. So in this example or method, we have our genome. We bisulfite convert the genome. Same things happen, single-stranded, fragments. So we'll take one of these fragments to demonstrate the principle. And you basically use random primers. So hexamers will bind to the converted DNA. This is part of one of the Illumina adapters here. So when you extend, you incorporate that sequence, and then a second random priming event allows you to generate an amplifiable library, and then these primers come in and complete the adapter sequence for our final PBAT library. Okay? So this technique is okay, depending on your application. The input mass is much lower than the previous libraries, but you get very, very biased coverage. Okay, so Illumina have a kit to do this. Was that me? And actually, they kind of take advantage of the hexamers and they target them towards CPGs. Okay, so they kind of enrich or over-enrich for CPGs in a whole genome context, if that makes sense. You get very high duplicate rates, though, and therefore very low diversity. So finally, that brings me on to post-PS libraries. So it's similar to the PBAT in the sense that we have our genome again. This time we fragment or mechanically shear the DNA before we convert it. Okay? So we bisulfite convert the sheared DNA, which becomes single-stranded and further fragments. Now you might think, well, why do we shear our genomic DNA before bisulfite conversion when bisulfite conversion fragments DNA? Well, if you look at the range of sizes in bisulfite fragments, they range from maybe 200 base pairs to 2 kb. So that's a large range of sizes, okay? Many of which are too large to sequence. So all of those large fragments we will lose as when we go to put them on the sequencer. We won't see any data from them, so it's a less efficient way. So what we do is we shear the DNA to a particular size, and then so a narrow range of sizes, and then we bisulfite convert, which just reduces those sizes a little bit. Okay, but we still get that tight window of sizes for optimal sequencing. So the method that we've been developing with a collaborator essentially adapts a single-stranded piece of DNA in a ligation-free, template-independent way. Okay, so there's no ligase involved in this step at all. It's quite efficient, so most of the fragments that can be adapted will be adapted. We then prime off this and extend. So we kind of got a library with one of the adapters. And then we do the classic double-stranded ligation and PCR amplify to get our final library. So one of the advantages of doing this is that when you ligate a normal fragment with your two Illumina adapters, they can go in at various orientations. Okay, so you lose some fragments because you've got two of the same adapter, for example. But here you won't get that, or you'll get less of it. So, of course, I'm going to say advantages now. you need a much lower input mass when you do this. I think Swift, I don't think he's here, but he may talk to you about this later. But Swift Biosciences have a kit which I think can go down to 1 nanogram of DNA. I think even less, and still get a good yield. You get a low duplicate rate because you're not random priming. You're not breaking all of your library fragments, so you're not losing them all during the prep, which means you get much higher diversity. And when you sequence, you get much more even coverage than PBAT. Okay? PreBS can give quite nice coverage as long as you start with enough DNA. Okay? So some example studies now. So this was whole genome bisulfite sequencing, not oxidative bisulfite sequencing, just normal bisulfite sequencing. And it came out of Joe Ecker's lab in the Salk Institute. So they took, I think, 18 tissues in 4 individuals. They did whole genome bisulfite sequencing. They sequenced to a minimum of 30x and up to 80x. So this is really deep data, which is good for bisulfite, but very expensive. So there are not many studies that go so deep. They looked at regions of the genome in all of these different tissues and kind of found some regions such as this, which I think is in aorta, where they see a different methylation pattern. So it's another DMR. They then looked at, so here are the tissues again. They then looked at cytosine methylation in non-CPG contexts, and they found that depending on the tissue, it can be quite high. Okay? So although they don't mention brain here, these are neuronal cells, so they must have done some neuronal sequencing as well. And as a percentage of the methylation, it can get to about 9 or 10%, so it's quite significant. And it also associates with non-proliferating tissues, again. So there's a bit of overlap with HMC. Now, when they looked at the context, so this non-CPG could be CPA, CPT, CPC. They find that the methylation is present mainly in CPA sites. Okay? So another study that was done, and this did use OxBS, was basically looking at cocaine addiction in mice. Okay, so mice were exposed to cocaine. Yeah, I know. Huh? I don't know. I don't know. So what they did some RNA sequencing, and they found that, remember the TET enzymes, the 10-11 translocases, the ones that oxidize the DNA modifications? Well, they found a decrease, a small but significant decrease in TET mRNA. So they thought, okay, well, let's look at HMC, because MC is oxidized to HMC. And globally, they found no difference in the levels. Okay, so they thought, well, maybe there's something a bit more regional going on. So any region that showed a change in HMC, they represent here. So more than half of those changes are actually in gene bodies. So they're not in upstream promoters. Okay, this is something a bit more interesting and not, let's say, the classical way of looking at things. So then they looked a bit more closely, and what they found was hydroxymethylation at the exon boundaries, okay, was associated with alternative splicing. So where you see an increase in HMC, you see an increase in the expression of a transcript with that exon that has HMC, okay? And the reverse is true, a decrease shows a decrease in the expression of a transcript that has that particular exon that's hydroxymethylated. So that's the first time that HMC was shown to influence alternative splicing. But then they looked at whether this was a stable change. So 1 month after the mice had been treated with cocaine, they again looked at the mRNA. They found, compared to non-cocaine treated mice, an increase in some mRNA transcripts in genes of interest to them. And these are genes that are implicated in addiction. They looked at some CPG sites with...
What tissue is that from?
This is from brain, specifically neural accumbens, I think, which is an addictive, or it's associated with addiction, I think. So CPG sites were looked at, and they see these increases in HMC levels compared to normals. That's demonstrated here. So this study has shown two things really, that it's implicated in alternative splicing, which I think is like really interesting, and it's stable. So it's stable for at least 1 month, which is pretty cool.
Did they know that the methylation is, which is coming first here?
Sorry?
Is the methylation leading to the differential splicing? Did they show that? Or do we know, or is it
Yeah,
possible
that's a good
that
question.
cocaine is leading to some upregulation of something else that's leading to this change
Yeah.
in splicing?
No, it's a good question. I mean, if TET1 is underexpressed, maybe it's... I mean, you'd think that if TET1 was overactive, you'd see more HMC, okay? But actually, it's underexpressed. So there is something interesting going on. But if you look at the paper, they don't address this. So finally, the last option for methyl sequencing is target enrichment. So a common way of doing this is amplicon bisulfite sequencing. There are some panels, so Nimblegen and Agilent, like their exome panels or targeted panels. They do exist for bisulfite. However, their performance is, I think, questionable. But what we do here is we bisulfite convert our genome again, we get single-stranded fragmented DNA. We design a primer to target our region of interest. We extend. We get another primer, extend again, and then when we cycle, we get copies of our region of interest. So you can do this on many regions, in many samples, but it's quite laborious, quite time-consuming. You're limited really by the number of PCR reactions you want to do. You can pool these in equimolar amounts. You can then adapt them. You can sequence them on the Myseq, for example. Or you can design these primers to have the adapters incorporated, so you do it in one step. And this is a very common way to look at very few regions of the epigenome. So now I want to talk about how we think about whole genome bisulfite sequencing conceptually, and how we are trying to improve it. So one thing that's becoming increasingly important is library diversity, okay? So these are all of the Illumina HiSeq platforms. The oldest is the 2500, and we come up here to the X10, so the HiSeq-X's. The numbers of reads is increasing here. So a read from an Illumina sequencer comes from one cluster, and one cluster comes from one molecule from your library, okay? So the more reads we're generating, the more molecules of our library we need to put on the flow cell, okay? So we have our genome, which I've represented by these colors. So these are the regions of the genome. If we make the perfect library, then these regions would be represented equally, okay? But in the real world, what happens is we're really good at making library for some regions and really bad in others, but in this example, it's just we've got loads of duplicates here, okay? So then what we do, if we want to sequence this library, have you guys loaded a machine yet? Are they going to load a machine? Okay. So you get a library, you make a massive dilution, okay, and then you put it on a flow cell. So really what you're doing is you're taking a very small sample from a very large number of molecules. If you put that on a HiSeq 2500, maybe you sample these molecules from our library. So when you sequence that and you count the duplicates, you get a fraction of about 12.5%, all right? But if we put this on a bigger machine, a better machine, we need to put more molecules on the flow cell because it can generate more reads. So what happens is we put a few more duplicates on, duplicate fragments, and from the same library, we get a higher duplicate fraction, okay? And then things get even worse when we go to the CEGXs. We end up with a huge fraction. So library diversity, the number of unique molecules in your library.
I can ask a question, Matt.
Yeah.
So with respect to the CEGX and bisulfite sequencing, the software does not work very well. So are you guys actually sequencing CEGX?
Some of our customers are, yeah.
And how are they getting that work?
We don't know. So with the CEGX, there was, I think the version 2 worked. Yeah. But version 2 chemistry and the version of the software that went with that, I believe worked. But when they went to 2.5, that's when everything tanked. But there are 2 sites that I know of, actually 3 sites.
So Harold's working at New York Genome Center, he's getting this to work supposedly, right?
Yeah.
And then UK guys are working over at Sanger?
I can't say anymore.
And,
I'm sorry. But what?
Illumina's in Cambridge too, right?
Well, yeah, but we're not with Illumina.
Still left over there.
One thing I have heard discussed is actually combining. So 50-50 mixing with genomic. So spike in.
50% 5x in this too, right?
Yeah.
That's crazy.
But you don't want to do 5x because you're just going to waste, right? So if you just do a genomic library, then you can do that.
So might as well just put it on just another system, or is this for run costs is cheap?
Well, yeah, exactly. So your per base cost is much, much cheaper. That's all right. So this is just a kind of schematic, conceptual idea, but this is actually real data. So we've got PBAT libraries, we've got pre-BS libraries, and we've got post-bisulfite libraries here. This is a curve. Have you seen a curve like this before? Okay, so there's a program called Pre-Seq, which will measure your library diversity. They call it library complexity. I call it diversity, but anyway. So these are the number of unique molecules. This is the amount that you're sequencing, essentially. So this dashed line represents the perfect library. All right, so let's say you sequence 800 million reads. Well, you have 800 million unique fragments. Okay, it never happens, right? But it's the ideal. So you can see with PBAT libraries that this line plateaus very quickly. So there are only, I don't know, maybe 200 million unique fragments in that library. So if you sequence, no less, sorry, there are only maybe 75 million. So if you sequence 400 million reads, all you're doing is just sequencing duplicates all the time. So pre-BS performs a little bit better, okay, with 1 microgram of DNA. With our prep, we can outperform with only 100 nanograms. 250 performs almost as well as 1 microgram, so we think 250 nanograms is a decent amount to put in. Less, and it affects your diversity, okay? But if you've got limited material available, then you might want to try.
So the gain of increase at the end, I mean, it's like, it's expensive to get that extra few percent.
Yeah, exactly. Yeah. So the other thing that you can do, well, I've just argued that you kind of want to, well, an increase in reads, numbers of reads results in more duplicates, right? So one thing that, most people when they do bisulfite sequencing, they do 2 by 100 base pair reads. Okay, why? Because their libraries are small. Okay, so if you want a 30x genome, you need about 100 gigabases. That means you need about 500 million reads, right? If you could do 2 by 150, which is the CEGXs do, as standard, you need a bigger library, but you can do it in 333 million reads, okay? You get the same gigabase output in fewer reads. So we have, as I said, fewer reads, fewer duplicates, but longer reads also results in a higher mapping efficiency, which with bisulfite libraries is really important because most of your Cs become Ts, which means you're essentially aligning to a 3-base genome. So it's much harder to uniquely align your sequencing data. So this prep that we've kind of developed with a collaborator is shown here. So we've got kind of 500-ish base pair libraries which enable 2 by 150 sequencing on the CEGX.
What's the efficiency of conversion then?
Of conversion,
Yeah,
of what do you mean?
bisulfite conversion.
So we see more than 99% conversion and less than 5% or around 5% MC to you, right, over conversion.
How do you get the large fragments? Is that just a scientific thing, or it's a pretty caustic reaction, isn't it?
It's a what, sorry?
It's a very caustic reaction, right? So it's pretty harsh to the other. So
I didn't hear all of that.
it's a really caustic reaction, right? So it's
Okay.
harsh on the DNA. So typically we end up with small fragments. So how do you guys get the larger fragments?
It's a good question. So it's a combination of shearing before you do bisulfite. So you have a narrow range of fragments, and then when you do the conversion, you keep that narrow range, but you just shift it smaller. All right. So that helps with the efficiency of the prep. But essentially, you know, bead-based cleanups, etc. You can get there. So these are the typical bisulfite libraries, which are kind of 300 base pair average. All right.
And those are the same input material amounts, yes?
You will see. You will. No, that's a good question. So the yield here would be lower for the same mass, okay, because you're throwing away smaller fragments. That's true. But this is something that we're kind of developing and testing all the time, and as the prep becomes more efficient, and the yield becomes higher. But a lower yield will mean lower diversity. So it's all about finding the balance between what you're trying to achieve.
So this would not be a 100 nanogram library, this would be 250 or 500 nanogram.
That was probably, yeah, it would be, let me think, 250. Okay, so 500 into the conversion, you lose about half of it when you convert, and then 250. So I'll happily take any questions that you might have. I'll immediately got one. I'll also talk about, or talk to anyone about any potential projects that they're interested in running with methylation sequencing, which we could do in the bar later or wherever.
How
So whole genomes, no,
Yes,
right.
so the core facility in my institution wants to charge me as much as $6,000
Yeah.
per sample
Yeah,
for whole genome bisulfite sequencing.
yeah.
Can you talk a little bit about the cost between the options that you presented to us?
Well, that's a good question. So, I mean, things like RRBS, you're sequencing 1% of the whole genome. So in theory, it's 1% of the cost, right? I guess MeDip is something fairly similar. I'm not actually sure, if I'm honest. I mean, amplicon sequencing is the cheapest option, but you need to know where you want to look at. I mean, everybody says the same, right? Whole genome's too expensive, and it's true. But it's only going to get cheaper if we make tools that are good enough to do sequencing on the larger machines, because that's where the cost per base really comes down, you know. But if you've got some kind of project, we can talk in more detail later if you want about your options.
So what's the big major part of the cost of sequencing, or the library prep? I mean, it's like having broken down. I mean,
But is it 6,000 from DNA to data? So they'll do the sample prep and everything? Yeah. I mean, the sample prep's cheap.
yeah.
I mean, really cheap. It's the sequencing. It all goes in Illumina's pockets, you know? That's where the money goes. Anyone else? Well, if you do, is it 19,000 a year on the X10? It's $1,000. Yeah. Yeah.
I didn't fully understand the post-BS, the process that you go through.
Yeah.
So you mentioned that it was. You guys.
If I can find it, yeah. Here. This one.
Yeah. So you
I just get to the end of the slide, but yeah, go on.
put on this red one
Mm-hmm.
in a ligation-free
Mm-hmm.
way. So how does that work?
I can't share that, unfortunately. This
Like a ligation-free ligation.
it's 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 a ligation-free adaptation, we can say. So this, as I said, this is something that's still in development, really. So this is not something that I can share.
Template independent? As in it goes.
So it's not preferring specific fragments. Okay, well,
Adaptive ligation events?
no.
And so how do you avoid having one grey molecule with two reds on it?
At both ends.
Yeah, is that possible?
So it's N specific. Okay.
Yeah, okay. How is it specific?
I honestly I can't. I wish I could, but I can't yet. So in the near future, I hope to. All right.
Thanks.
But Swift to do something similar, I think.
We'll find out in a minute.
Yeah. Ask the Swift guy, you know. Well, that's the problem. Yeah. Yeah.
It feels like bisulfite sequencing, since we don't actually know what most of these do, and there's not really a code, and it seems like it's rather random changes in expression, that you would have to pair this with some sort of like real information. Like this is real information, but like pair it with like end changes, like with RNA.
Yeah. So it's becoming, for those that can get the money, increasingly common to do multi-omic studies. So, you know, we've got some customers that have done RNA-seq, whole genome bisulfite, ChIP-seq. Okay, so they're layering all of these data together. And it's really only then that you can start to infer biological meaning, okay? But it's expensive still. So, I mean, you're right, you need to keep it in context. So, actually, one of the interesting things about just doing bisulfite and not oxidative bisulfite is when you try to match that data with transcriptomic data, it doesn't really match. Okay, there are loads of problems. OxBS can help there. because you're just looking at just methylation. If you do both together, which, you know, it isn't two reactions, so it's another expense, you know, it can help even more, because then you're getting more to exactly what MC is doing and HMC is doing when you compare that to the transcriptome, right?
Does the HMC follow that rule where if it is there, then there is decreased, is it decreased expression?
Increased.
Increased expression?
Yeah, that's what it's associated with so far. I mean, there are new studies all of the time.
Does it have to be in a particular area in the gene, or?
So, I mean, I don't know whether it has to be in a particular area to drive expression. I'm not sure about that, but it's generally found outside of promoters. So it's more associated with gene bodies and enhancers.
So they're not in CGI?
No. Well, they can be, but not in CG islands. No, sorry, yeah. They can be in CPG sites, right, but not in the islands. So one of the most common ways of looking at methylation is using an array. Okay, it's the 450K array from Illumina. But the content of that array is very promoter specific. But they're bringing out a new array soon, which is kind of more enhancer, gene body focused, which hopefully will be more interesting for HMC studies. Any other questions? No? All right. Thank you.